So good afternoon, ladies and gentlemen. It's my great pleasure to introduce Professor Mariya Ptashnyk from the Heriot-Watt University in Edinburgh. So Mariya studied first in Lviv in Ukraine, and then she moved to Heidelberg for PhD, and then she had a couple of postdoc or lecture positions at the WTH in Aachen, also in Heidelberg, and then she was a lecturer at the University of Dundee. And finally, she arrived to Edinburgh, and now she is a full professor there. So Mariya's research interests include biomechanics and biomathematics, I would say, and also PDEs, including stochastic PDEs. And from the talk about modeling or biomathematical modeling is also scheduled for today.
Yeah. Thank you very much for invitation and for the introduction. It's a great pleasure for me. I was for many years trying to come to Prague, and finally I managed having several friends here. It's nice to see all. And I will present here some relatively a bit old and a bit recent work, but it's the active research areas that now I'm interested in on the coupling between the chemical processes and mechanics and growth, and specifically in the biological tissue. And here I will present this on the looking on the mechanical properties and growth of the plant tissue, but I believe that the framework is relatively general, so it can be also applied to the other biological tissue, to the human tissues, and specifically now, like maybe motivated also with the situation in Ukraine, I'm quite interested also looking on the bones biomechanics in the connection with the developing the modern protheses for veterans. And there will be three parts. If I might not manage the last one on the locally periodic microstructure, but I will definitely try to consider the modeling of the plant cell wall and explain and motivate why we're considering the specifically concentrating starting with the plant cell walls and after then the plant tissues, and then first just the biomechanics, coupling between the biomechanics and chemistry that happening in the cell walls, and then extending this to include also the growth. And why we are interested specifically in modeling of the biomechanics of plants, we're specifically interested in the cell walls because they are like a skeleton for the plants. Yeah, plants doesn't have any bones, and everything is dictated with the anisotropic properties and the mechanical properties and structure, anisotropic structure of the cell walls in order to have all those beautiful shapes and flowers and stable trees. And more specifically, I was interested to see and to model and better understand the coupling between the microscopic structure, the mechanics, and the chemistry, what's happening starting with the cell wall and after then extending to the plant tissues. And what we know from the biology of the plant cell walls that we have very stiff microfibers, which was built from the cellulose, which are embedded in the relatively soft cell wall matrix. But in the cell wall matrix, we have a lot of chemistry, and specifically the creation and breakage, like dynamics of the so-called loading crosslinks. And here in this specific research results, I will concentrate on so-called calcium-pectin crosslinks. And it was motivated by these biological experiments that when they were looking, this is control plants of the Arabidopsis, how they're growing. And when they were perturbing this calcium-pectin crosslinks chemistry, the interaction, which I will introduce in a moment, how this is happening, and specifically by perturbing, they overexpress the inhibitor of the pectin metalloesterifiase enzyme, specific enzyme, which controls the dynamic of interaction between the calcium and pectin. Then you have this disturbed growth, this strange wavy growth. And actually, if you increase the overexpression of the inhibitors, then you can even get like the tissues of the roots becomes almost like resolved, and you have like a mass which are not properly growing. And this was somehow like motivated by this experiments. We were concentrating on those calcium-pectin crosslinks, but there are many different other crosslinks and dynamics, specifically also hemicellulose and interaction between the microfibers, cellulose microfibers, and hemicellulose in cell matrix and so on. It means that if somebody is interested in the whole dynamics, and there are more different points that we can look. And then in so-called microscopic modeling, when I'm concentrating, deriving the model on the level of the individual microfibers, I will distinguish between the mechanical properties of the microfibers, which is cellulose, and then the mechanical properties of the matrix, and then the dynamics of the calcium-pectin crosslinks inside the matrix. And then the coupling between the mechanics and the chemistry will happen in that those crosslinks is changing mechanical properties of the cell wall matrix. This means that I will assume if I have higher density of the crosslinks, then my matrix becomes more rigid. It's less extensible. From one point, this is one-way coupling, so the elastic properties of the cell wall matrix will depend on the density of the crosslink, and the another way coupling will be that the mechanical forces can break those crosslinks. Yeah, they extend and then break. It means that then you decreasing by applying the forces or by developing the forces in the cell wall, you decrease in the density of the crosslinks. And just briefly how this calcium-pectin crosslinks chemistry happens is that you have two forms of pectin, metallosterphite and metallosterphite. Don't worry about remembering this. But somehow the cells imposing into the cell wall pectin in the metallosterphite form, then you have the enzymes of removing the metal groups, and then they become metallosterphite, and then they can bind with the calcium. It means that you have like pectin chains. Two pectin chains will be bound with the calcium in between and create those crosslinks and like crosslinks chains, which gives you additional stability. Okay. And this is the biological background. And now for the modeling, we assume that a cell wall material is hyperelastic. It means that we consider the momentum equation, and also to simplify the idea, and also having that the extensibility is relatively small, we assume also that it's that we can describe this, but by linear elastic, the equation of linear elasticity, where I distinguish between the elastic properties of the cell wall matrix, depending on the crosslinks density, which I will denote by B3, and the elastic properties of the fibers, which embedded in the matrix. And this is like small, like a cartoon of the piece of the cell or representative part of the cell wall. And also we consider also that the cell wall matrix can be also viscoelastic, again, just the linear one, and then so the viscoelastic properties will also depend on the crosslinks density. And this is the coupling between the chemistry and elasticity, so the elasticity depends on viscosity depends on the crosslinks density. This is for the part of this, when we consider just the cell wall as isolated material. When we will consider the whole plant tissue, then we will consider the poroelastic material, because you have also what is one important mechanism also, which is controlling also the stability and all processes, is the water flow in the plant tissues, and also we have very high pressures in the plant cells. It means that then we for the plant tissue we will consider the equation of the poroelasticity to describe the mechanical formation and the water flow. And now for the whole calcium pectin chemistry, of course, we have like the two different forms of the pectin, metallosterphite, demetallosterphite. We have the crosslinks, we have the enzyme and the calcium molecules itself. I just combine them all in the vector. And just to give the main ideas, that now we can model this the chemical interaction by reaction diffusion equation, where then the reaction term becomes on it's dependent on the deformation or displacement gradient, and we will consider that it depends on the positive part of the trace of the stress or strain. Okay. This means that what is now mathematically challenging is the direction terms. It's a nonlinear function of the displacement gradient, and this is actually when looking on this problem from the point of view of nonlinear PD. This is the only actually challenging point, because in the linear elasticities we know how to deal with this, especially if you can assume nice properties on the elasticity tensor. Of course, this dependence on the cross-link density gives you a bit of technicality, because you need to prove that the density is converge strongly when we're looking on the analysis or on the Galerkin approximation. But if we have the reaction diffusion equation, then this is relatively easy to show. And the only challenging point is really to deal with this nonlinear dependence on not on the deformation. This will be easier, but it's the deformation gradient or the displacement gradient. Okay. And I was saying that we're looking like on the multiscale analysis because, and distinguishing between different properties of the cell wall matrix and fibers, because they have very different, means that in the elastic modules and the Young modules between the matrix and fibers is 4 order of magnitude difference. And the other natural microstructure is that we have thousands of fibers in the cell wall. It means that we have that elasticity tensor, if you distinguish between the elastic properties of the fibers and matrix, is very highly oscillating function. And this is where the whole multiscale analysis, the homogenization, what I will present here, is coming into the play, because since you have this high oscillating coefficients in your equation, to simulate this is very challenging. It's even challenging for one unit cell, because you need really to resolve on the surface very fine, because you really have high differences between the properties. And then if you have 1,000 of them, then this is almost impossible. And the idea is, okay, can I now apply some multiscale analysis techniques? There are some effective equation which is well defined on the homogeneous part of the cell wall, but will take into account the fact that I have this difference in the properties of the fibers and the matrix. And this is what is actually will be the main idea of this part of the work, to derive those macroscopic equations. And what I will present here, looking on the periodic distribution of the fibers. This is the easiest one, and this is like applying periodic homogenization, if somebody is familiar with with those ideas. But in general, what we can do also mathematically rigorous is to look on so-called locally periodic. And here is like the cartoon of the locally periodic. The idea is that you have a patches or layers of the microstructure which are periodic, that you can represent, or that you can approximate. Like, for example, here you have each layer of the fibers is rotated, but it's possible to approximate this microstructure with the unit cell, which will go diagonally, as already sketched here, so you can represent this by a locally periodic medium. So you will have the patches with the periodic microstructure, but with the unit cell, which is not corresponding to one fiber, but to the fiber which is going into the direction of the rotation, or actually depends on the derivative of the rotation angle. And you can approximate and then also apply the rigorously locally periodic homogenization techniques and derive the macroscopic equation. Or random distribution, when your distribution is some ergodic stationary process. And there's also, if you have some tails, there are some techniques also possible, but I was looking only on the nice ergodic stationary. And then you can apply the tools of the stochastic two-scale convergence for the random, and there are also tools of the Karl measure, which allows you to show also the convergence on the surface of microstructure of the random distributed microstructure. And then for the periodic and locally periodic, we have two-scale convergence on unfolding methods, which allows you to analyze and derive the equation rigorously. But I will just, for the simplicity, I will show you the periodic case, and then if I manage with time, then at the end I can give you a few ideas of deal with the locally periodic one. And then here I just briefly state, okay, this might be too short and too fast, just to mention what those two main methods that I will use is the two-scale convergence on unfolding operator. And the idea of the two-scale convergence is like a bit generalized notion of the weak convergence in LP. And the difference is only that it takes into account that you know that you have microscopic and macroscopic variable. The macroscopic one is the one that describes the process on the whole homogeneous tissue, and the microscopic one which describes the variations on your fibers on the cell, on the surface of the cells. There are those local variations. And then, as you will intuitively think that then the limit should be also the function of two variables, macroscopic and microscopic. And this notion of the two-scale convergence was very much motivated by the formal asymptotic expansion, which we know that, like, assuming that you have the dependence on the x and x over epsilon, and you're doing Taylor expansion in the small parameter epsilon, then you will obtain this for the in the expansion if you're doing the ansatz. And why this is helpful, because this gives you a bit more information on the weak limit, because weak limit will be just the average of since the weak limit will be just of this sequence, because sequence will be still bounded in LP, and the weak limit of the U epsilon will be just the average over the microscopic variable of U of X and Y. It means that this gives you a bit of more information, but the most important thing, what this gives you is that this highly oscillating coefficient that we have changed between the properties of the matrix and fibers on the scale of the size of epsilon. This means that you have 1 over epsilon in your definition of the function. Those highly oscillating functions, as we know, they converge only weakly in the standard LP, but they will converge strongly into scale sense. And this helps us to deal then with the limit in the PDs, because usually the gradient or the symmetric gradient will converge only weakly or weakly to scale when we go, but then the coefficient will converge strongly, and then you can deal with the strong weak limit. Otherwise, if you take just the standard convergence, then you have only weak times weak, and you don't know where you are. This is for the two scale. And for the unfolding method, it has the same idea. Also, again, you know that you have this macro and micro coupling, and what this operator is doing, especially, is useful when you have so-called perforated domain, when you're looking on the domain between the cells here, on this domain with holes of the size epsilon, or if you're looking on the so-called oscillating boundaries of your microstructure, then the unfolding operator is mapping the function defined on this epsilon-dependent domain of perforated domain or the boundaries of the microstructure into the domain which is fixed, to the function which is defined on the fixed domain, but automatically dependent on both variables, macroscopic and microscopic. Yeah, you have the rest scale boundary or the rest scale domain Y, E, and then the macroscopic. And then it's somehow like you can work now with the standard convergence in the double variable domain, which allows you to use all technologies and methodologies that you know from the standard analysis. It's only that where you have to pay attention that with this applying this unfolding operator, you're losing any regularity with respect to X. because in X you always piecewise constant, and the whole regularity goes in Y. This means that this is where you have to be careful. But for example, if you want to show strong convergence of the sequence, then it's often easy because you can apply, show the Cauchy properties in a fixed domain. Otherwise, if you would like to show the Cauchy properties in the gamma epsilon, then you will have, like, you don't know what to compare, because then your sequence, subsequences living on the different domains. Yeah? This is the only technicality which helps to derive the, especially if you need some strong convergence results. And now just the main ideas for the analysis, that first, since we have this two-way couplings, that we have the elasticity tensor depending on the Clausius density, and we have also the reaction diffusion equation for the pectin chemistry depending on the displacement gradient. We use in the fixed point argument, and the only way where it's a bit more technical is that we need to show this fixed point, consider the fixed point operator in L infinity. It means that because for the since the elasticity tensor depends on the cross-link density, you will have the contraction inequality in terms of B in the L infinity. You're not getting anything better. And this is only where you really have to play with all this Gallardo-Nuremberg inequality and Moser-Alikarpos iteration techniques. It means that here is like a lot of technicalities to work with in order to ensure that you have these contraction properties in the L infinity. This is only the non-standard part, which is dictated with this two-way coupling. And then having those well-posedness and also a priori estimates, which automatically get in when you show in the well-posedness, and applying two-scale convergence and unfolding operator, you can derive the macroscopic equation. What I was saying is that now I want to overage and have that I have some, again, the equation of the linear elasticity or viscoelasticity when we have here additional viscous term for the cell wall matrix. And then the reaction diffusion equation defined now in the whole. G is now the whole homogeneous domain without seeing fibers separately. But now my information that I had initially, the fibers which had different mechanical properties in the matrix, is embedded now in so-called homogenized elasticity tensor. This means that now I have a new elasticity tensor in my homogenized equation, which see the properties of our original microstructure, which is now rescaled to so-called unit cell, is now of order 1, not of order epsilon. And how to compute this means that to compute the macroscopic correctors, and this again comes from the convergence of the gradient in two-scale. This means that this converge. Any derivatives in two-scale limit converge to the normal derivatives, as we would have from the weak convergence, plus the derivatives of the additional function, which is then play the role of so-called corrector, or which gives you the information how the overage mechanical properties influenced by the mechanical properties of each of the components of your mixed medium. And again, here a bit what is maybe not so standard classical is that we will have here dependence of the elasticity tensor on the cross-links densities, means that this will depend not only on the so-called microscopic variable of the rescale microstructure unit cell, where we can consider fibers in different directions, but this will be also depending on the time on the X on your homogeneous macroscopic domain. It means that in each point of your macroscopic domain, you will have to zoom out and solve the unit cell problem to determine the effective elasticity properties. And this is usually computationally quite expensive. What the people doing this using or reduced-based model method, or coupling between the coarse and fine grids and resolving, for example, the determining elasticity properties on the coarse grid and then computing the macroscopic equation on the fine grid. This is what we will do for the growth. But when we have, when we can assume that the cell wall matrix is isotropic, then from the experiments on the pectin crosslinks chemistry, we know that elasticity tensor can be as a fine function, as the linear function of the Young, written as the linear function of the Young modulus with two elastic tensors E1 and E0. And then also from the experiments, we know that this function of the Young modulus is just, again, a linear function of the crosslinks densities, means that actually at the end, we had to compute only elastic properties for two crosslinks density and then just use the linear interpolation. And this was allowing us to compute the, like to have a cheap computation. Otherwise, you would have to resolve or looking on the different resolution of the grid. And what we also looked at, for example, if the crosslink, if the fibers are not just parallel, but rotated across the cell wall thickness, you can do the locally periodic homogenization that I was mentioning here, but here, but here what we did also that we just consider the fibers in one direction, then rotated across the thickness using the rotation angle for the homogenized elasticity tensor. And this give us also ideas how this rotation of the fibers affects the displacement, because when especially on the plant roots growing, due to the growth, the fibers are rotated, and it was assumed that actually this is one of the mechanisms why the cells stop growing. So when they rotated through the whole thickness, at some point the displacement is so small that they might not be growing anymore. But this was just the assumption. But if doing the computation, what this was quite interesting that we see that if we consider parallel fibers, it means that they are all parallel to the or in one direction parallel to each other and orthogonal to the z direction, and z direction is along the length of the cell. And we apply the forces F at this end of the tissue or part of the tissue. We have no displacement in z direction on this one, representing some symmetry. And we have some turbo pressure, which is denoted by P1 inside of the cell. And we see that if we have the rotated fibers across the cell wall thickness, then the displacement in the z direction is one order of magnitude lower, which just means that gives you a bit of, which was intuitively clear that if you have rotated fibers, then the displacement will be lower than if you have all parallel, but this gives you also the direct quantitative information. And if you have the viscoelastic material, then doing the only like the idea what we have to do like technically to prove the all multiscale analysis results, we need to first regularize with perturbation of the in the time derivative, and then go to the limit also in the regularization parameters. Technicalities. And now I would like to present the second part where we also consider the growth of the plant tissue. And again, we have the microstructure now given not by the cell wall fibers, but we assume that, okay, we have already some properties of the cell wall, and the microstructure now is given by the distribution of cells, yeah? Because we will then consider the structure and properties of the cell. And then we have some turbo pressure inside of the cell. And again, we assume that the cell walls are hyperelastic material. And to model the growth, we consider the approach proposed by the Rodriguez, Hocker, and McCulloch in 1994, where we assuming that in the growing biological material, we can assume the multiplicative decompositions of the whole deformation gradient is decomposed in the product of the elastic and the growth tensor. And then for the elastic, we can consider the standard momentum equation. And for the growth, we have to define the dynamics of the growth depending on the biological situation. and looking, and especially to have the growth rate. In our case, we will relate this again to the stress or to the strain. And this approach was used in the many different settings, considering also plasticity, remodeling, and for the plant growth, there are a couple of few results, a few works when they were looking on the plant tissue growth, but more on the macroscopic level, and also the recent work on the surface growth. So now for our multiscale setting, what we consider, as I was saying, that we have the momentum equation for the elastic deformation, and we have pressure inside the cells, which is given by the called turbo pressure, which is driven by the water inside the cell. And we consider that maybe on some parts we have some directly boundary condition, or we have some forces on the part of the domain, and just applying forces on the other part of the macroscopic boundaries. I mean, macroscopic is the external boundaries of the tissue. And for the dynamics of the growth, it's motivated by the well-known Lockhart law, where we say that the rate of growth is proportional to the stress when it's big, and we have the growth when the stress is larger than the given threshold. Yeah, this means that it's in some sense phenomenologically defined. And now, again, we have the microstructure given by the size of the cell, since in any biological plant tissue, we have, again, hundreds or thousands of cells. This means that the epsilon will be a small parameter which defines the ratio between the size of a cell and representative size of the tissue. And again, for the elastic deformation, we consider the linear elasticity with some elastic tensor, which can depend on the microscopic structures, means that we can distinguish between different elastic properties of different parts of the cell. And for example, also middle lamella, which is gluing the cells together. And here, maybe just to emphasize, when we consider the doesn't go back. Oh, what I have go back. Here, yes. When we consider the multiplicative decomposition, it means that then the elastic, the deformation gradient will be the full deformation times the inverse of the growth. And then when we linearize, in order to apply the linear elasticity, when we linearize then the strain to consider the linear strain, then at the end what we have the symmetric part of the elastic displacement, and then also the symmetric part of the growth and minus identity. So that this is also important to, that you're not ending up only with this part, which you might be naively will think, but if you're doing the linearization of the total strain and just ignoring the quadratic term of the displacement, then you have these three components. For the turbo pressure, we also represent that in order to consider that we have some macroscopic variation of the turbo pressure across the tissue, but it's constant inside of each cell, which is known biologically, we have also to decompose the turbo pressure in the macroscopic one and the microscopic one, which is depends on each cell. This is also where you need to, from the modeling point of view, pay attention. And now for the growth, what we are assuming is that we are not assuming just that it depends on the stress or strain, but also biologically motivated, but even more technically motivated. There are some technicalities. What we assume is that the growth depends on the overage or the local overage of the cell, of the stress or the strain in the cell wall for each of the representative cell. Okay, it means that we are overaging locally on each of the cell, and this tells us about the growth of individual cell and then in total of the tissue. And what this will end up in the limit when we derive in the macroscopic equation for the growth of the tissue, the growth tensor will be a macroscopic tensor. Will be dependent only on the macroscopic, on the average variable of the whole tissue, but not on the microscopic one. And also, this setting also allows us to obtain the generalized Corn inequality, because in generally what we have this f, the growth tensor will be just a bounded tensor, and if it's just a bounded, we have this very nice results by Neff and Pompey, and the generalized Corn inequality doesn't hold. But the fact that we have that is bounded, but it's also constant on each unit cell, and also that we have that is a growth. So this means that the determinant is larger or equal than 1, and also the eigenvalues of the tensor larger or equal than 1. Then for the u, which are in the space, I forgot to add here, that is in the space of the, which is orthogonal to the rigid motion, we will have that the symmetric gradient of the gradient of u multiplied with this tensor f, with those properties, can be is controlling the H1 mod. Yeah? But this is really, we need both. We need that this uniformly bounded from below and above and constant on each unit cell we using, and we also using the fact that this determinant is larger or equal than 1 and eigenvalues larger or equal than 1. Otherwise, in general, it's not correct. Yeah? It's not true. And there are nice control example, I think, in this work by Pompey in 2003. And having this one, then we can derive all a priori estimates, and again, using fixed point in the space of the corresponding growth tensors. And having the a priori estimates, we apply the notion of the two-scale weak convergence. Now, in order to pass to the limit in the also in the symmetric gradient and we need the strong convergence of the growth. It's also where we need this macroscopic behavior of the growth, this averaging properties of the growth in order to hope to have some strong convergence for the growth tensor. Then we can go to pass to the limit in this transformed or in the product of the displacement gradient and the growth. Because for the displacement gradient, we cannot have any strong convergence. And then having those all convergence results, we can then again derive the macroscopic equation, which is now, because originally we set up everything on the properties of the individual cell. Now we derive the macroscopic equation, which will be defined on the macroscopic level of the part of the plant tissue when we have the elastic deformation and growth, and the growth depends on the elastic stresses or strengths. And what we obtain as we would have from the normal homogenization of the equation of linear elasticity, that we will have a new elasticity tensor, the homogenized one, as we have similar before. But now also because of the macroscopic pressure that we have imposed inside of the cell, given by the pressure of the water, we have in the homogenized stress now also this additional contribution of the pressure. And this reflects very nicely the actual poroelastic nature of the plant tissue when we have them pressurized by the water, because this is what we will have. Of course, in our case, the P1 is prescribed. It's not as in the standard poroelastic equations that you will have coupled with the dynamics of the pressure, but this term is similar as in the Biot equation. And then the microscopic pressure becomes just as a reaction as the force term in the equation. And then for the growth equation, we will have the new macroscopic stress, so the growth rate depends on the macroscopic stress, which you have, as I say, those two components, the elastic one and the one which comes from the pressure. And as before, that the elasticity, new homogenized elasticity tensor is given by the solution of the unit cell, which represent the microscopic structure, and also has reflects also the growth, yeah, because we just pull back on the reference domain. It's also you have this inverse tensor of growth. And then for the coefficient, which is corresponding to the pressure, we will have a similar additional corrector and the macroscopic coefficient, which also has depends on the elastic properties of the tissue. And now with the looking at the macroscopic equation, we can simulate and look different properties and how the homogenized elastic properties depends on the growth. And here, in this situation, we just have prescribed growth by given that it's diagonal, and we have no growth in the y direction, and just growth g, the factor which is varying in the x direction, and then we see that elastic, the Young modulus in the direction of growth will be increasing, but decreasing in the direction orthogonal to the growth. The Poisson ratio is increasing with the growth, and shear modulus is non-monotone function of the growth, where the coefficient which coming from the pressure, the one in the direction of the growth, in the absolute value becomes smaller, in the orthogonal is larger. And it also gives us the idea that we consider also different shapes, a bit elongated cells, and can look on the structure, how it's the shapes of the cell depends on influence the elastic properties of the tissue. Yeah. And in general, this macroscopic equation also they allow us. So, for example, here we can already consider some variation in the shapes of the cells because we pull in back in the reference domain and look how those macroscopic variation in the shape of the cell influence the elastic properties of and the behavior and the growth of the tissue. It means that, because sometimes if you have some weird changes, for example, of the thickness of the cell wall and so on, you might geometrically not represent them in order to solve you numerically, use your model numerically on that strange shapes, but you can vary those properties in the macroscopic equation and look on how they affect the macroscopic behavior. This is just one of the advantages, and also we just to check that our multiscale analysis is consistent. We also perform the error analysis by simulating microscopic model defined on the level of individual cell, and simulating the macroscopic model defined on the level, the homogenized one, and then we see that with the increasing number of cells, which correspond to the decreasing epsilon, the decreasing of the small parameter, we have that the error is decreasing, which we would have expected. And here also representing the algorithm, what I was saying that one of the way to have efficient simulation, since on each point of the space, we will have to solve those unit cell problems in order to determine the macroscopic elasticity tensor and coefficients in order to determine macroscopic growth. In order to have this more efficient, we consider the fine grid and coarse grid, and on the coarse grid, on the nodes of the coarse grid, we define the unit cell and the macroscopic coefficients and properties, and then solve the macroscopic equation on the fine grid, and then iterate. And this gives the efficient algorithm. How much time do I have?
That's 15 more minutes.
Sorry?
15 more minutes.
15 more minutes. Oh, okay. Then I can present also the last part where they are saying that, okay, we are not restricted to, even for the rigorous mathematical analysis, we are not restricted to the periodic microstructure, but we can also do locally periodic. And this was motivated by another biological material by the lobsters, because they were looking at the properties of the lobsters are very stable, but they're also very light, the exoskeleton of the lobster. And what the biologists or engineers see is that you have two layers in the exoskeleton with different rotations of the chitin fibers. One of them also called slow rotated, and this one I schematically represent with this, that you have the layers of the same direction, then you're rotating them, and this is like so-called fast rotating, when each layer of the chitin fibers is rotated in the different directions. And they are rotated usually across the axis, which is orthogonal to the exoskeleton. And then in order to deal with that type microstructure, I was thinking that we need something like locally periodic setting. And in general, it means the locally periodic that you can hold your tissue, you can split in the patches or layers, which can be of the order epsilon, but to the power r when r is strictly smaller than 1. Because if the r is equal 1, then you have each layer or each patches of the size of your microstructure, but r between 0 and 1 gives you that you will have several periodic cells or fibers in the same patch in the same periodicity, but in the next patch you will have different periodicity. Of course, those changes on the periodicity needs to be smooth, nice. You need a Lipschitz function of the deformation. And here, as we see, okay, if the angle of I will like for this setting, I will have angle gamma, which will be C2 function of x3 of the third direction, because it's rotating only across the third axis. And then I have these patches of the order of the size epsilon to the power r, and I can choose any r you can have from the experiments, for example, and then we can apply directly here the locally periodic two-scale convergence unfolding method to derive the macroscopic equation. In this case, since each layer is rotated in the different angle, if the rotation angle is C2, then I can define a new unit cell, new periodicity, in such way that I can consider a patch of the size epsilon to the power r, but then there will be a restriction on r. R needs to be, I think, between 2/3 and larger than 2/3 and smaller than 1. It's still smaller than 1, it means still a bit of space. But in those patches and those modified periodicity, I can approximate those locally periodic microstructure will be well approximate this microstructure, and then applying them locally periodic two-scale convergence, I can derive then the macroscopic equation. And just the notion of the locally periodic two-scale convergence is very similar as we had for the periodic one. It's only that now you have periodic one in each of the patch. And for that case, since you consider in the test function which will be periodic periodicity depending on x, it means that you don't have a fixed periodicity, but now you have to approximate them and define so-called locally periodic approximation, so that you consider the function which are periodic with a periodicity d of x and epsilon in each patch n, yeah? This means that then you somehow you approximate and you look using this locally periodic approximation. That locally periodic approximation, if epsilon goes to 0, will be converging if the function is continuous, and the microscopic variable will converge to that function. This means that in the limit, even if you choose in here locally periodic approximation, in the limit you will recover the correct the test function you choose, and you get the limit function. But it gives you the whole properties of your microstructure of the original problem, which is embedded in this locally periodic approximation. And then when you consider in the elasticity tensor, which will be vary between the fibers and the matrix, then that elasticity tensor will be a locally periodic approximation of some function which is has the X-dependent periodicity. So this is somehow like placed all together. And then in the macroscopic equation, you get in again the same structure, since that the structure of the equation doesn't change. It's only that now the elasticity tensor, again, you have also the macroscopic new elasticity tensor. And you see only that in your unit cell, how you define the macroscopic elasticity tensor, again, the same philosophy. It's only that you see this rotation matrices in your in the transformation of your unit cell. This is when you have those slow rotated, like you have particles of the same periodicity, and then if you have this fast rotated that you need to approximate, and your unit cell will depend on X. This means that now in each point of the macroscopic domain, you will have different unit cell problem defined on the different geometry of unit cell, because it now depends on the rotation angle, so on the derivative and on the cosine. This means that because you somehow, your periodicity needs to change in the direction of the rotation. And I will stop here, just give you the ideas. And then what I was trying to present here is to develop a framework which is coupled mechanics and elasticity, and also the mechanics and growth. And the next step would be to also incorporate the remodeling and the changes in the mechanical and chemical processes driven by growth, and also to develop the so-called surrogate models that you can, because sometimes those multiscale problems are quite expensive, and you want like to train your neural network or machine learning methods on the nice fine simulation, finite element simulation, but then had some good approximation that you can run them very fast, specifically if you're working with the precise experiments and you have to determine the parameters in your model using the data. Then this was the idea also to combine this with the machine learning methods. And thank you very much for your attention.